Good morning. Yeah, this talk really does follow on, not from the technical aspects of whole genome sequencing, but the fraught area of databases and sharing information. The work that I'm going to talk about is part of the same Listeria research program that Phil talked about. It's a different work stream, and it's devoted to trying to create a database that collects data and sequences isolates to provide new data for a Listeria whole genome sequence database. These are pretty fundamental issues, and a number of people have raised them already this morning. The Food Safety Research Centre engages with industry for all of its projects and also with government, and so we need to work closely and find a way of sharing and collating the data while respecting privacy and avoiding risk. So whole genome sequence databases provide really good opportunities for getting more data, getting more value out of the data which is being produced at an increasing rate. A database or a shared database provides the opportunity to create a consistent format for managing the data, and also to provide quality metrics that gives assurance that the data is solid. It gives opportunities to share the data between both contributors and users. We can create unique and anonymous identifiers, and we can be flexible about metadata. Metadata is the additional information beyond the sequence information that is information about the source, the timing, and the history of the isolate itself. We can compare isolates to detect similarities and linkages. We can create phylogenetic trees using these standardized visualization tools that you've already seen. For this particular project, the Listeria workstream, our key interest was really to try and get a handle on the population or the genetic population structure of Listeria across all sources in New Zealand. That's our ultimate objective by bringing all this information together. The other really important function of a database is it provides context for a new isolate. And this speaks to one of the issues that Pierre raised about the potential for misinterpretation of data. The more background we have, the more context we have about what an isolate means, I think that's a benefit. But obviously there are a number of risks and issues. Who controls the database and is responsible for uploading new data? What's the database structure and software? How do we control access and privacy? What level of metadata is appropriate? How do we go about securing permissions to add data? And what do we do about unexpected linkages, which a couple of people have already spoken to, where we're looking at isolates from different sources. And we're really trying to integrate information here from a variety of sources of the isolates, clinical cases that are identified from human health surveillance, food, water and environmental isolates, and we'd also like to be able to collate and compare information from international databases. So what have we been doing in this project over the last year or so? We've created a prototype database for Listeria whole genome sequence information. The data held includes minimal metadata, as well as multi-locus sequence type assignments from standard genome assemblies. We've created a user interface and also a database structure with a really minimal metadata set that was agreed with various contributors. And quality control metrics are being added. At the moment, the database is hosted on an ESR production server, which enables access by outside parties. At the moment, the only outside parties with access are partners within the Food Safety Research Centre. We've created a system for controlling access and privacy using a tool called the Research Electronic Data Capture, or REDCap. This is a web-based application. It was developed in the States originally for clinical research, and it was designed to provide a secure environment so that research teams can collect and store highly sensitive information. We're adding visualization tools, standardized visualization tools to the database, and we're negotiating with sources of isolates and or data to add more information to the database. The project has resources not only for collecting data and adding it, but also for collecting isolates and then sequencing those within the project. So what have we been doing so far? We're adding whole genome sequence information from about 400 historical clinical isolates of Listeria, and this has been negotiated with the Ministry of Health. These isolates have been sequenced by ESR, and they're not concurrent isolates. There's about a 6 to 12 month stand down period, so these are historical. This negotiation with the Ministry of Health is in parallel with a process that the Ministry is undergoing to provide sequence data from clinical isolates into international databases, NCBI, which is increasingly becoming standard practice for public health agencies around the world. Public Health England, Canada, the US are all uploading clinical isolate sequence data at the moment. We're also adding whole genome sequence data from food industry isolates that have been negotiated principally by Plant and Food Research, and we are in the process of sequencing some historical isolates from previous MPI projects undertaken by ESR, and that's been negotiated with MPI directly. So as I said before, currently only Food Safety Research Centre partners have access to this information. This is just a screenshot of the user interface. As you can see, there is really minimal metadata here. We are only identifying the sample type as either clinical or non-clinical. That's the level of detail for the source. We give the year, we give a serotype where that's appropriate, and then the rest of the information is the MLST sequence data. The database does not hold raw data reads. That information is held elsewhere, but the database would provide an opportunity to access that information for deeper analysis. We can create tree diagrams using similarity metrics like MLST loci or SNP analyses, and these are being embedded in the database. So the challenges that we are still working through, and I emphasise this as a work in progress. At the moment, who controls the database? At the moment, ESR is the curator of the database, researcher access only. The structure and software is using the REDCap tool, and within REDCap we can create data access groups that maintains record sets by contributor, and we have the opportunity to add a record level embargo code that constrains access according to the contributor. The level of metadata that we're including, as I said before, is really minimal. We have a unique identifier, we describe the species, the country, whether it's clinical or non-clinical, the year it was isolated, and the typing data such as serotyping. Any additional metadata is held only by the original contributor. We're continuing to negotiate permissions to add additional data. The owner of the isolates or the owner of the data can specify who can access their data, and that option is available within the REDCap software. As far as unexpected linkages, they're largely of historical interest. This is a legal aspect that I think we still need to work through. Phil mentioned that linking an isolate with a case that was 5 or 10 years ago would be very difficult legally to prove, but I think this is an aspect of the database that we still need to work through and get legal opinions. It's important to stress that the database is about understanding the genetics and the population structure of Listeria. It's not an outbreak investigation tool, it's a research tool, and as such, any future addition of information from particular projects under the research centre, we'll conduct a risk assessment about the risks around the whole genome sequence information that will come out of that project. So where to from here? We're going to create additional rules and options for access by outside parties and whether or not they might be required to contribute to the database. We need to increase the access for industry partners in MPI. We want to add more Listeria isolates, but we've really spent a huge amount of time working through these issues, and it is taking a long time as people get more comfortable with the concept of contributing to these databases. But once we've worked through these issues, we're hopeful that in the future this template can be used for additional pathogens for research centre projects. We're adding visualisation tools and also channels to access the raw data for deeper analyses. So just to conclude, this database approach is it's 12 months work and a lot of talking to various agencies. It's not finalised yet, we've still got some issues to work through, but we have this issue in common with other countries, Australia obviously, and I've also talked to people in the US who are wrestling with this issue. But I think the benefits of having this information together in a standardised database are substantial, and I think we should work through these issues and realise the advantages of combining information. And if we would welcome any feedback on the approach that we're taking and from industries who might be interested in contributing or using the database. So, thank you.